Okay, Assalamu alaikum. So, if you remember, last time we had done the graph of the effect of substrate concentration on the rate of enzyme catalyzed reaction, and under that we had studied the importance of Vmax and Km as being indicators of how the reaction is taking place. And to sum up, I can say that Vmax and in Km are two separate entities. It is not a must that if an enzyme has a high Vmax, it also has a high Km. They just tell us about the efficiency of an enzyme. There can be enzymes, as you can see in this table. I think we studied this table last time, which can have a high Vmax and a low Km at the same time. So these are two separate entities which are giving us different kind of information for an enzyme catalyzed reaction. To sum up, we can say Km is that substrate concentration at which the reaction velocity is half its Vmax and half the active sites are occupied. If any enzyme has a low Km, it means the affinity of that enzyme for the substrate is high and vice versa. And we can see that in this table. And now we move on to just a summary of these two properties of enzyme catalyzed reaction, which means that we can compare the enzyme efficiencies by taking into account the turnover rate and the Vmax, right? The turnover rate is the rate at which the enzyme can convert any substrate into the product per second, and we saw that the turnover rate of carbonic anhydrase molecules was very high. It could actually break down 600,000 molecules of carbon dioxide in a second. And theoretical possible Vmax means that that enzyme which is completely saturated with substrates, which means all active sites are occupied. So to sum up, we can say that the rate at which the reactions occur can be determined by two factors. It can be determined by the affinity of the enzyme for the substrate, and it can also be determined by the Vmax. So this was about it, and then we move on to this is also, I think, the repetition in which I would be just reading the last two points, which is enzyme with a low Km means it has a high affinity for the substrate, and it also means that that particular enzyme जिसका low Km है, it needs a lower concentration of substrate to reach Vmax than an enzyme with a high Km. यानी अगर हमारे पास दो enzymes हैं, one with a low Km and the other with a high Km, so the advantage of the enzyme which has a low Km will be that it needs a lower concentration of substrate to reach Vmax. अगर उसको Vmax पे पहुंचाना है तो it needs a lower concentration of substrate as compared to enzyme with a high Km. ठीक है, इसका मतलब ये बिल्कुल भी नहीं है कि if an enzyme has a high Km, its turnover rate is small. It can have a low Km just like carbonic anhydrase, and at the same time its turnover rate can be very, very high, meaning it can actually break down just like carbonic anhydrase 600,000 molecules of carbon dioxide per second. सिर्फ इसमें यह है कि उसके पास substrate concentration ज़्यादा होनी चाहिए, and in case of carbonic anhydrase the substrate is CO2. As soon as the CO2 starts accumulating inside tissue cells, this enzyme becomes very active and it can remove 600,000 molecules of CO2 in a second, but it has a high Km. ठीक है? And now we move to again summarizing the same thing, the same discussion that we did last time. I'm repeating it so that this concept can be reinforced. Enzymes with low Km, enzymes are more likely to be saturated with substrate in the normal conditions within a cell. और इस तरह के enzymes का फ़ायदा ये होता है कि if inside the body the substrate concentration varies, अगर कोई variation हो रही है तो it will have less effect on the rate of formation of the products. यानी कि substrate concentration की change will not affect the rate of formation of the products in case of enzymes which have a low Km. So they have an edge, they have an advantage. On the other hand, the enzymes with high Km are less likely saturated with substrate. ठीक है, और अगर variation होती है in the substrate concentration, it will have a greater effect on such enzymes. So this means that the enzymes with low Km can adapt better to changing situations within the tissue cells or within the blood plasma, whereas the enzymes with high Km, they are less adapted, because if there are slight variations in the substrate concentration, it will have a greater effect on such enzymes, meaning कि इस तरह के फिर enzyme जो हैं वो अपनी product, the rate of formation of product will be less, right? So that's why the enzymes with low Km have an advantage. You can be given such kinds of graph in exams in which you might be asked, एक आपको स्टैंडर्ड बता दिया जाएगा कि इस तरह का ग्राफ आपको बनाना है, and you have to plot a graph, annotate it and explain it. Graph number 1, in which we have an enzyme 1 which has a low Km. So how will that graph look like? ठीक है, कोई enzyme है जिसका low Km है, तो how quickly can it reach Vmax? How quickly can it reach half Vmax? You have to show and then you have to annotate and explain in the graph. और उसके बराबर में एक दूसरा ग्राफ बनाना है about another enzyme which has a very high Km. तो वो अपने Vmax पे कब पहुँचेगा? Where will that half Vmax lie and why is that so? तो आप generally किसी भी point पे दिखा सकते हैं. For example, in this case, if I have to show an enzyme with a low Km, तो since this is the substrate concentration x-axis, तो मैं कहीं पे भी दिखा सकती हूँ. For example, I can show the Km over here. So this will be the half Vmax. ठीक है, अगर ये half है तो फिर यहाँ पे कहीं पे आएगी Vmax and then I can show के ये rough sketching है कि this enzyme achieves half Vmax and Vmax earlier than this enzyme जिसका Km आप कहीं यहाँ दिखा दो because Km ज़्यादा है. So then this means that the half Vmax will be somewhere here and then Vmax will be somewhere here. So कहीं पे भी आप दिखा सकते हो roughly to just tell the examiner that you know what is the meaning or what will happen to the Vmax or the rate of an enzyme catalyzed reaction in an enzyme with a low Km and in an enzyme with a high Km. So this is how these graphs should look like. You can see that the graph in blue, the enzyme 1, is that enzyme which has a low Km. ठीक है, let me just highlight it for you. This is the blue graph, ठीक है, and it's ending over here and you can see it has a low Km. तो low Km का मतलब it reaches half Vmax also earlier and Vmax also earlier. ठीक है. So this is how you will annotate and explain your graph. आपने graph पहले roughly sketch किया and then you annotate and explain it. Annotation means making short explanations. So how have I annotated it? For blue graph, I can say the enzyme with a low Km is saturated with substrate and works at a fairly constant rate. So hence variations in substrate concentration will have less effect on this enzyme. Whereas the enzyme with the high Km, जो red वाला graph है enzyme 2 का, it has a low affinity for the substrate because it has a high Km. याद देखें Km इसका ज़्यादा है and so it has a low affinity and needs a higher concentration of substrate, half Vmax, Vmax, forms fewer enzyme substrate concentration in the same unit time as compared to the enzyme with the low Km. And एक और चीज़ भी है कि the enzymes with the high Km जो red वाला ग्राफ है, in them the active site is a less good fit for substrate. ये ज़रूरी है लिखना इसलिए कि when you say the enzyme has a high Km, इसका मतलब ही ये है कि that enzyme has a very low affinity for its substrate. तो क्या मतलब हुआ इस बात का कि इसकी affinity low है? It means कि the active site of the enzyme is a less good fit for the substrate. उसी less good fit होने की वजह से we say the affinity is low and hence the Km is high. तो उसकी वजह से क्या होगा? Fewer enzyme substrate complexes will be formed in the same unit of time and such a reaction needs a high concentration of substrate to achieve the same effect. Why? Because the enzyme has a low affinity for the substrate. So this you are supposed to answer this question in the similar manner as it has been depicted in this graph. And then just to give you an idea कि किस तरह के questions आ सकते हैं, तो for example I am just sharing this question with you and I'll be reading it and explaining it to you at the same time. Some students investigated the effect of substrate concentration on the rate of reaction catalyzed by an acid phosphatase enzyme A. ठीक है, x-axis पे the concentration of substrate is plotted. On y-axis, the rate of reaction is plotted, right? And you can see a typical वो जो graph आपका आता है जो initially increase हो रहा है, then the rate is slowing down and finally it reaches a plateau. Let's see what the question has to say. The students investigated a different phosphatase enzyme, enzyme B, and found the value of Km to be higher than 0.3 millimoles per decimeter cube. ठीक है? अच्छा, एक है एंजाइम ए जिससे स्टूडेंट्स ने ये एक्सपेरिमेंट किया एंड दे गॉट दिस ग्राफ, ठीक है? एंड देन दे आर डूइंग एन अनदर एक्सपेरिमेंट जिसमें एक दूसरा एंजाइम ले रहे हैं, एंजाइम बी, एंड दे फाउंड ऑफ द Km फॉर एंजाइम बी टू बी हायर दैन 0.3 मिलीमोल्स पर डेसीमीटर क्यूब. ये क्यों कह रहे हैं 0.3 मिलीमोल्स पर डेसीमीटर क्यूब? क्योंकि मोस्ट प्रोबेबली 0.3 मिलीमोल्स पर डेसीमीटर क्यूब मस्ट बी द Km फॉर एंजाइम ए. सो वी कैन इवन calculate and see it, because ये के if this reaction is stopping somewhere here तो ये इसका Vmax होगा, ठीक है? this line. This will be the Vmax. If this is the Vmax, then we can calculate the half Vmax. Exactly, हम half calculate कर सकते हैं, और जहाँ पे भी half Vmax आएगा वहाँ हम नीचे इसको ऐसे प्लॉट करके देखेंगे तो हमारे पास Km आ जाएगा. So the students investigated a different phosphatase enzyme B and found the value of Km to be higher than 0.3 millimoles per decimeter cube. Explain the difference between the values of Km for these two phosphatase enzyme. तो we have two enzymes, the enzyme A जिसका हम Km ग्राफ से निकाल लेंगे, ठीक है, in millimoles per decimeter cube, and the enzyme B Km has been told to you which is more than 0.3. ठीक है? So let's say if this is half Vmax, Km for enzyme A, and then the Km for enzyme B is more. So they are asking you to explain the difference between the values. क्यों है ये different? And the marks are 2, right? Let's look at the answer. The same points that I mentioned on my graph on the previous slide. Enzyme B, why is the Km for enzyme B higher? ये आपसे examiner पूछना चाह रहा है. तो आप कहोगे कि the enzyme B has a high Km. Why? Because it has a lower affinity for its substrate as compared to enzyme A. The higher the Km, the lower the affinity of the enzyme for its substrate. So that's why the enzyme B needs a higher concentration of substrate to reach Vmax, half Vmax. Enzyme B is forming fewer enzyme substrate complexes in the same unit of time. Enzyme B active site is a less good fit for substrate. So if you note down these points, कि less good fit है, एक unit time में कम enzyme substrate complexes बना रहा है, it has a lower affinity. So these are the same points which were mentioned on this slide, right? So these kind of technical questions you should have a practice and go through past papers and see if you can practice them more as well. And then after doing this, we move on to the topic of enzyme inhibitors. Okay, the name tells you a lot. These are compounds that inhibit the enzyme activity, right? A knowledge of enzyme inhibitors is very important because it enables a biologist or a researcher to have an in-depth knowledge about how to design a drug. Because if you keep in mind antibiotics, which are meant to kill bacteria, so if I can have a knowledge about how the different kinds of enzymes, what their structure is, and if I could create medicines or drugs or design drugs which can actually inhibit an enzyme that is responsible for protein synthesis inside a bacteria, or an enzyme that is responsible for the bacterial cell wall synthesis. If I could actually make an inhibitor of that enzyme, I will be successful in designing a drug against many microorganisms. So the knowledge of enzyme inhibitors can enable me to do so. Then so many chemical reactions, metabolic, anabolic and catabolic, are happening inside my body, which are a part of my homeostatic mechanism. So the knowledge of enzyme inhibition can give me an in-depth knowledge about how homeostatic mechanisms are operating, what are those factors that result in a positive feedback or a negative feedback mechanism. और अगर किसी में कोई metabolic disorder है, like there is diabetes or there are certain other metabolic problems, they can be understood by having an understanding of how enzymes work and how they can be inhibited inside your body. कोई problem होगा जिसकी वजह से enzyme inhibition हो रही है और जिसकी वजह से homeostasis is not occurring properly. For example, diabetes mellitus is a disease which is an imbalance of the homeostatic mechanisms. So a knowledge about enzyme inhibition will help a medical researcher or a doctor to understand such metabolic disorders in a better way. Enzyme inhibitors also enable us to design antidotes for poisons or toxins, right? Because we in the ERs or the emergency rooms, we very frequently see that people have been poisoned accidentally or deliberately by any kind of a poison, right? By a toxin or maybe any other reason. If we don't know how that toxin inhibits an enzymatic pathway inside your body, we will not be able to save that person's life. For example, आपका snake venom है. Snake venom causes sometimes instantaneous death. There is one snake venom that as soon as it enters the blood, it causes or stimulates the clot formation. So this means it actually stimulates your clotting cascade pathway. तो अगर आपके पास ये knowledge है, अगर फ़ौरन से आप उसको एक ऐसा enzyme inhibitor दें जिसकी वजह से clotting activate हो रही है किसी patient में, and he might die, so you can save the life of the person. So this means the knowledge of enzyme inhibitors is very, very essential, right? And yes, enzyme inhibitors, the knowledge of them will also enable us to study how tissue damage occurs due to toxicants. तो अगर हमें समझ आ जाएगा तो हम उसकी remedy भी ढूंढ लेंगे, right? So this is about an introduction to enzyme inhibitors. And okay, the same thing. And then we move on to the different kinds of enzyme inhibitors. Inhibition or enzyme inhibitors can be studied in two ways. We can say that these are chemical compounds that can inhibit an enzyme reversibly and irreversibly. And then यानी enzyme की inhibition reversible तरीके से हो सकती है and irreversible तरीके से हो सकती है. How many kinds of inhibitors do we have? These chemical compounds, we have competitive inhibitors and non-competitive inhibitors. ठीक है? To understand competitive and non-competitive, I will first share the diagrams of both of these because the pictures tell you a lot, right? This is the diagram of a competitive inhibitor. So it shows you the enzyme which is yellow in color and the substrate which is green in color. In the second diagram on the right hand side, it shows you that there is a competitive inhibitor, a compound whose shape is very much similar to the shape of the substrate. ठीक है? जिसकी वजह से क्या हो रहा है कि instead of the substrate occupying the active site, the competitor occupies the active site. In this scenario, if the substrate concentration is less, the competitive inhibitor gets a chance, because uska size same hai, it gets a chance to fit inside the active site of the enzyme instead of the substrate, and hence the name competitive inhibitor. So there is a competition between the inhibitor and the substrate for getting access or binding to the active site. जो भी पहले जीत जाए. So if the substrate concentration is low, the competitive inhibitor will get a chance. Whereas if the substrate concentration is high, the competitive inhibitor will not get a chance to inhibit this enzyme. So such kind of an inhibition is a reversible inhibition. Why? Because it can be reversed by increasing the substrate concentration. So this is how it is. For example, I can show you this slide. ठीक है? You can see that there is a substrate who needs to bind to this active site, and there is a competition between this substrate and the competitive inhibitor whose shape is similar to the substrate. So when the substrate concentration falls down, the competitive inhibitor gets a chance to bind to the enzyme, and look what's happening. Here the competitive inhibitor binds to the enzyme, and here it's not able to bind to the enzyme. So such kind of a situation has two alternatives, either this alternative or this alternative, right? Acha, what is the daily life example? The daily life example is that of the antibiotic sulfonamide. Okay? Just like penicillin, sulfonamide is also a class of antibiotics. The structure of this antibiotic is very similar to a compound which is known as PAB or PAB. Okay, this substance is needed for the growth of pathogenic bacteria. तो हम क्या कर रहे हैं? We are actually playing around by synthesizing the antibiotic sulfonamide and sort of customizing the structure of sulfonamide in such a manner that it is very same to a substance which is needed for the growth of pathogenic bacteria. Bacteria require PAB, this substance, to live. Since sulfonamide structure is very similar to this substance PAB, therefore the sulfonamides, they bind to the enzyme and they inhibit PAB. So this is how it is. I can show you a detailed description over here as well. Yes. See, what does the textbook say? The knowledge of competitive inhibition helps us to understand the effect of a group of antibiotics known as sulfonamides. Antibiotics destroy infections without damaging host tissues. Sulfonamides were the first antibiotics to be used and were developed during the 1930s. Okay? They are very similar, yani sulfonamides are very similar to a structure or a compound which is known as para-aminobenzoate. This substance is essential for the growth of many disease-causing bacteria. The bacteria need this compound for the production of folic acid, an important enzyme cofactor. तो अगर हम sulfonamides देंगे, तो sulfonamides will act by inhibiting the enzyme that is needed for the synthesis of folic acid from PAB. Now, as AS students, you don't need to go into so much detail कि sulfonamide किस चीज़ के लिए चाहिए और वो चीज़ किस चीज़ के लिए चाहिए. Simply you can say, if you are asked about, write down a named example of a competitive reversible inhibition, you will say the example is that of sulfonamides. They are used to kill pathogenic bacteria. Their structure is similar to a compound known as PAB, which is required for the growth of these pathogenic bacteria. So long as sulfonamides are given, they will competitively inhibit the substrate which is needed for the reaction to be completed and the growth of pathogenic bacteria to be completed, which is why as long as sulfonamides are given, the bacteria will not be able to grow. Itna kafi hai isko samajhna, right? In fact, if I go back to that slide, I think this much is enough. Structure of sulfonamides is the same as PAB, a substance needed for the growth of pathogenic bacteria. The bacteria need PAB to live. Sulfonamides are similar to PAB, therefore they bind to the enzyme and inhibit its synthesis. So I think it's enough for today, and then in the next lesson we will be discussing about non-competitive inhibitors. Allah Hafiz.